Welcome back to the course Fundamentals of General Immunology, and in the last module we looked into detailed aspects of mucosal immunology. We understood that how this mucosal surface, particularly to the gut, lung as well as genitourinary tract, is important in terms to fight with anything which is entering into this area. Particularly these are more prone because these are open areas. We also understood about the proper way in terms of tolerance, immune tolerance, oral tolerance, and other kind of central and peripheral tolerance, which we understood in earlier lectures as well. We also looked into how these tolerance can be utilized for the clinical setup and see if they can be utilized to do treatment for the patient who are actually suffering from autoimmune disorder such as multiple sclerosis or some other conditions which are untreatable because of the host self-immune response. So therefore what we understood till now is mucosal immunology is very, very interesting, and therefore there are many other facts which are there, and in addition to that there are beautiful thing happened in this area of research where last year the Nobel Prize is given to very specifically discovery of the peripheral tolerance, or in general the tolerance research was identified as one of the pioneer kind of research in 2025 Nobel Prize winning in 25. So we will try to see some of the interesting facts about mucosal immunology, and let's try to move on and see that what are the important facts about the mucosal immunology, and also we'll try to see the specific say contribution of the mucosal immunology towards the tolerance and the award of the Nobel Prize in 2025, and where 3 scientists were given collectively the Nobel Prize for discovery of tolerance. So that means this area is really fascinating, and it is attracting many researchers across the world to do certain research in order to find out some cure about autoimmune diseases and some of the things which are flaring up in the human society in recent years. So let's try to see one by one some of the facts which are there. So mucosal immunity constitutes the largest part of the human immune system. That means this is very, very a large area which they are covering, far larger than the skin. So since the gut wall, the respiratory tract and everything is filled with this mucosal epithelium, so therefore these are constituting very large area, even larger than the skin, because skin is considered as the largest organ in the human body, but these are larger than that. Most of the antibodies, actually about 80% of the body antibodies are produced mostly by the mucosal immunity or mucosal immune response, particularly the gut lamina propria. These are the largest antibody producing organ because the gut wall itself is largest, and therefore we can consider that the mucosal immune response which are coming from the gut wall is maximum. Very interestingly, we looked into that IgA, which is an immunoglobulin A, exists in dimeric form, and this dimeric form is actually a secretory form of the immunoglobulin, so we called it secretory immunoglobulin A, and these are actually linked. The 2 immunoglobulin A are linked with the J chain, and these are actually the secretory component. So this dimeric form of the immunoglobulin A is about 15 times more potent than the monomeric form of the IgA, and therefore these makes them to be resistant against the degradation by the bacterial enzyme. So essentially the dimeric form of IgA is a very, very important form of the serum immunoglobulin, which are quite stable and can be stabilized in a multiple different way. Particularly they are not getting degraded by some of the enzymes which bacteria can secrete, while monomeric forms are prone. So their half-life and their degradation capacity are quite high as compared to the dimeric form. So therefore dimeric form is important. The other thing is regarding the microfold cell, which we have discussed in multiple times, where we have discussed microfold cells multiple times, where we have looked into how this antigens are processed from the gut wall, and they can processed and pass on to say lamina propria or other areas where they can be processed by dendritic cells and further process those antigens to the immune cells. So microfold cells are very unique cells of the alimentary canal, which are not present anywhere else, and therefore these are also very unique cells, and particularly they lack mucus and cilia. So essentially they don't have a typical structure of the alimentary canal cell. They are not secreting mucus, they are not having cilia. They are simply catching and processing the antigen, so they are very unique. The other important aspect about IgA is they do a phenomena called immune exclusion. What do you mean by immune exclusion is we yesterday also, in the last class also, we looked into the immune exclusion phenomena, where the IgA molecule simply mask the virus and bacteria basically, and because of the masking, the virus and bacteria won't be able to bind to the surface of the epithelial cell and unable to do infection. So therefore it's a masking phenomena. So essentially IgA can trap all the receptor present on the virus and bacteria, and therefore when the receptors are trapped, they are not able to properly bind to the surface of the alimentary canal or any other cellular surfaces, and therefore can be protected from the infection. So this is what we call immune exclusion mechanism. Other very important cells which are present in alimentary canal is actually intraepithelial lymphocyte, which is present in the epithelial area. These are the unique lymphocyte, and they are the first kind of responder to the entering pathogen. So even we don't need any sensitization of the adaptive immune system and all. These are resident kind of lymphocyte present in the area. So once they are trapping the antigen, they are encountering the antigen, they can do an effect immediately, and therefore these are called the primary responder or the first responder. Other thing is our unique aspect about is the presence of antimicrobial peptides. So we looked into multiple areas. The cells are secreting a small protein or a small polypeptide chains. These are called antimicrobial peptides. So we'll look into this antimicrobial peptide in a while. Some of those are very commonly known as, some of the very common examples are such as defensin, which are secreted by Pineth cell in the alimentary canal, intestinal cell, and these are very specific for bacterial cell wall. So we'll see how these antimicrobial peptide can act and destroy the bacterial cell wall. The other very important thing is the mucosal immune system is directly connected to brain, and therefore we see that the olfactory mucosa, the specific neurons which are present in the mucosal surface, can directly communicate with the brain. The very important example, very nice example, is during the COVID-19 infection, people who are infected with COVID-19 start getting a typical symptom where they were unable to get the smell. When the people are infected with COVID-19, they started saying that they are unable to get the smell and the taste of a particular food substances. That stands for the COVID-19 can directly damage the mucosal olfactory sensory aspect, and therefore people were not able to get proper smell or taste of that food particle, and essentially that suggests a phenomena that these can directly link to the brain. So brain and gut is actually brain gut axis people are looking into now very critically. So we can say that this unique aspect of alimentary canal and the gut mucosa along with other mucosal surface are making this system a very ideal system to fight with the pathogen. So let's move on and see what is the antimicrobial peptide and what are the different antimicrobial peptides which are unknown to us. So these are actually the polypeptide. That means these are the chain of amino acids, and they could be 100 and less number of amino acids depend upon what kind of antimicrobial peptides which we are talking about. Most commonly characterized antimicrobials are defensin and cathelicidins. These are the two antimicrobial peptides which we have studied a lot in terms of their structure, their function, and what they cause and how they are doing their effect we have looked into very critically. Apart from that, there are other antimicrobial peptides which are characterized, such as histatines and dermacidines. So histatines and dermacidines are actually present in the specific areas of the body. Particularly histatines are present in salivary gland, and also dermacidine is actually present in the skin area. So derma is actually related to dermis area. So these are actually the antimicrobial peptide identified from that area. Apart from that, we do have anionic peptide also. We'll see now that apart from this cationic peptides, we do have anionic peptides also. That means based on the charge of the peptide. So essentially, defensin, as I said, the defensins are mostly studied. There are two kinds of defensin, alpha and beta, and beta is actually rich in these defensins are rich in beta sheet fold like structure. So these are actually beta sheet structure. So these can be considered as parallel or anti-parallel. What we see here is anti-parallel because one is in this direction, the other is in the opposite direction. So these are anti-parallel beta sheet, and then again we do have some alpha helical structure. So these are all secondary structure of the protein. So defensin looks pretty much like this. There could be a structure deviation when we talk about the defensin coming from the plants. So this is kind of conserved protein. So plants also secrete multiple different kinds of defensin. Insects also secrete defensin. So these are very well established or evolutionary conserved peptides which are secreted from the vertebrates in order to protect them from the environmental stress or the pathogen. So these are some of the things which we need to understand. Now let's see how these antimicrobial peptides, which we are saying that can kill the pathogen, how they are causing an effect. So essentially, when we see this is typically an example of defensin. So defensin is actually an amphiphilic molecule where we have some of the portion which are actually cationic and some of the portion is actually anionic in nature. So these are, therefore they are called amphiphilic. So the general charge of the cell wall, general charge of the cell wall in bacterial cell is actually negative. So the peptides are generally cationic in nature, which can go and bind to this anionic surface. So negative charge of the bacterial surface, the cell wall, is mostly because of the ions or kind of groups which are present in gram-positive and gram-negative bacteria. If we look critically into gram-positive bacteria, we know that gram-positive bacteria contains teichoic acid or lipoteichoic acid, and this can cause the charge deviation in these bacterial species. While in case of gram-negative bacteria, we see lipopolysaccharide. So lipopolysaccharide or LPS can give a negative charge to the cell wall, and therefore these all material can easily bind to the positively charged molecules, and therefore defensin is an ideal molecule. So once defensins are present in the system, they can perturb the bacterial cell wall, they can penetrate inside, and once they are penetrating inside, since they are arranging themselves because of their own property, so the hydrophilic and hydrophobic areas can arrange in such a way that they can disrupt the cell wall in a very different way. So since these are positively charged, they can bind to the cationic lipid and then they can perturb internally, and accordingly the charged molecule can be maintained in such a way that they can do a porosity on the cell wall, and therefore this can rupture the bacterial cell wall, and therefore these cationic peptides are equally good for both gram-positive as well as gram-negative bacteria, simply because they can act on the charge-based biology. So these are actually biological membranes, so they can easily work and perturb the bacterial cell wall. So this is how the antimicrobial peptide acts and destroy the things into the from the system. So this is all about some of the things which are there, and let's try to move on and see what are the things which got acclaimed in the scientific community in 2025 related to mucosal immunology. So the Nobel Prize is generally given during the first week of October, the first Monday of October, actually. So in 2025, the Nobel Prize in Physiology and Medicine is given to 3 scientists collectively. And these are all from 3 different area research group. So Mary Bunko, Fred Ramsdell, and Simon Sakaguchi. So these 3 scientists got Nobel Prize for their discovery of peripheral tolerance. So they identified a very unique thing about how this tolerance can be produced. So Mary Bunko was from the Institute of System Biology, Seattle, USA. Fred Ramsdell is from a company, biotherapeutics company in San Francisco, while Simon Sakaguchi is from Osaka University, Japan. So these 3 scientists did a very pioneer experiment which we'll try to understand now. And since we are looking into mucosal immunology, this all contribution goes to how the mucosal immunology can perturb and modulate the immunity in the human being. So what Sakaguchi started realizing that what we know is actually when microbes are infecting the antigen presenting cell, they are processed through the T cell through binding by T cell receptor. So the antigen can bind to T cell receptor, which we have also looked into. There are DNA, germline DNA that have all the, so in the germline DNA we have all the information that can code for alpha chain and beta chain, and after this the alpha and beta chain can be rearranged to make a transcript which can form the protein. So T cell receptor is actually both alpha and beta type. Alpha is actually lacking the diversity region. We know VDJ recombination, so these are the variable loop, and then we have a constant region. So this constant is actually here and this variable is here. So the VDJ recombination can give variation in different kind of T cell receptor, and T cell R is actually of multiple types. So that was the thing, that there are variation in the T cell receptor, and T cell can bind to the antigen and give variety of different kind of immune response. So the experiment started with a very unique way which Sakaguchi did. So the very first experiment, or the pioneer experiment I would say, was when the Sakaguchi removed the thymus from the 3-day-old mice. So they removed the thymus, that means the T cell is completely aborted in those mice species. So these mice where the immune response was nullified because thymus was removed started getting autoimmune diseases because basically there are no T cell in those individuals. So what he observed, that when you remove the thymus, the mice is developing autoimmune disease. And suddenly he encountered a second experiment, and he realized the result from the second experiment, that from a healthy donor, if you can transfer the mature T cell to those mice who lacks the thymus, can become healthy. So something is coming from this healthy donor that is providing some kind of an protection from the autoimmune diseases, making this sick mice healthy. That means there is something which is related to T cell biology which was absent, and therefore the mice become sick and suffering from autoimmune disease, while the mice which received the T cell from the healthy donor become healthy, and therefore the autoimmune is actually related to something from the T cell biology. But what was that was not understood at that time. So he took those experiment critically, looked all the cells very specifically, that what are the things, what are the markers available in the cell, and if at all anything that makes sense. And he did back-to-back multiple experiment, and finally he came for a conclusion that whatever T cell which he inoculated into the mice, if you can only put CD4 specific T cell, the matter is not going to resolve. The mice is still sick. But when you are adding CD25 positive T cell population, then mice become healthy. That means there are something which is there in the cell that express CD25 that makes this mice healthy. That means if you give CD4 positive T cell, then this is not going to help. But when you inoculate something with CD4 plus along with CD25 plus, then the mice become healthy. That means they are protected from the autoimmune disease. That means he concluded that this CD25 is actually a very critical protein that is responsible for making these mice healthy. And he identified this as a subset. Now we know that these are called T regulatory cell. Of course, this is not the only marker which we have identified at that time. Now we know that there are multiple other markers such as FOXP3 that decide the T regulatory cell. But this is just a generated data that gave the Nobel Prize to these people. So CD25 positive cells are actually protecting. Now if you look back and see what is CD25 positive, so this is actually an IL-2 receptor. So if you have this cell that express this CD25, then essentially the IL-2, interleukin 2, can come and bind to this area. So this is binding, giving a docking to the IL-2. So these are IL-2 receptor essentially, which we now studied in great detail, but this was done long back, so at that time it was not known. So he has been given a credit for discovering this very specific cell signature that is allowing the mice to get them cured from the autoimmune diseases. Now, so this was the identifying feature which Sakaguchi did in his own experiments. Now, taking these data, two other scientists did some pioneer work, and the story started with this mice. So this mice is called scruffy mice. Now this is a wild type mice, and this is actually a scruffy mice. Now if we see closely the tail region, this is a tail of a wild type, while this is the tail of a scruffy mice, which looks diseased actually. So scruffy stands for the scaly condition of a hair in their body. So they are not having a healthy hair coating and all. So essentially this is a very good mice model to study some of the autoimmune diseases, because these mice are actually showing some of the autoimmune diseases. People were not knowing what is the reason behind that, but ideally if we do an sectioning and see the immunohistochemistry, we can easily see that there are issues with the cellular system. So we can see easily lot of infiltration of the lymphocyte in the site of inflammation, and also this is actually the healthy cell. That means the architect of the cellular architect is actually lost in this scruffy mice. So this gave an idea that essentially these all cellular architect and all leading to a condition where the scruffy mice is showing autoimmune disorder. Now the question was why it is so, although this is understood that this mice is actually bred through the lines and discovered in a very different way. People use these mice for all different purposes, but we're not knowing where the mutation was. So ideally these two scientists, Ramsdell and Bronco, look into very carefully all the genomic sequences of the mice chromosome and finally identified that the culprit is in the X chromosome. So the gene which is responsible to give this problem to the scruffy mice is situated in the X chromosome. So this is X-linked problem, and the gene was identified at FOXP3. So which is what we know now, fork head box protein 3 is actually the unique protein that gives the scruffy mice a specific phenotype which can lead to the autoimmune disease condition. That means this FOX3 mutation can cause a problem in the scruffy mice where the mice is suffering from disease condition because this gene is non-functional in a proper way. That means the contribution of this to scientists is to identify the actual cause of the autoimmune disorder in this mice species. So ideally we can see that the science developed from a phenotypic marker. We looked into the phenotype and then we tried to see what is happening in the genotype. It's kind of a Mendelian genetics where the Mendel can see what is there in the pea plant. Say the plant is green or yellow, if the pea plant is round or wrinkled, and then try to see what is there that decides this change in the phenotype pattern. So we called it forward genetics. We see the phenotype and try to see what is there in the genotype. Similarly here, we looked into scruffy mice that there is a mutation, there is a change in the phenotype. So what that change in the phenotype is actually deciphered in the level of genotype later on by these two scientists, identifying FOXP3 is the culprit responsible for doing this phenotype, and that's located in the X chromosome. So beautifully orchestrated that the Sakaguchi experiment tells that there is T cell involvement, and they also say that there is a gene which is responsible for this kind of a phenotype. Now collectively, what is inferred from these two findings, that suppose we have an endogenous protein fragment which are processed by the immune MSC1 molecule or 2 molecule, and these are endogenous protein fragment. So these are self actually, and if the T cells are generating the immune response against the self antigen, this is what will lead to the condition of autoimmune diseases. That means a T cell that has slipped through the test in the thymus, which we have understood in the earlier cases that central tolerance or the peripheral tolerance can allow the T cell to be eliminated by energy or by apoptosis if they are reacting to the self antigen. But in case if they are somehow slipped out and can react from the fragment of body's own protein, then this is not good. So to control this T cell overactivation, we have this individual cell called T regulatory cell. So if we can use the T regulatory cell, so these are patrolling cell actually. So these are intervention by patrolling regulatory T cell. So this T cell can control the overexpressed T cell, and therefore they can downregulate the immune response which was generated in the patient. That means the autoimmune response can be controlled by regulatory T cells. So that was the contribution that gave the Nobel Prize in 2025 to these three scientists. Now what is the output? What we can use this biology to see what is happening in case of human being. So what is very understood that we can upregulate the T regulatory cell population in case of autoimmunity. And if we have an autoimmune problem, we have to see how we can upregulate the T regulatory cell population. While in cancer, we wanted to control the T regulatory cell population. So we can downregulate the T regulatory cell population in cancer. Why? Because say if we can have a regulatory, then it can be immunosuppressive in nature. We don't want. So ultimately this can lead to immunosuppression. In cancer, you want to be having an immune response in order to remove the unwanted cells which are present in the human body. So can we use the T regulatory cell to treat cancer? That is what is the output of this whole research. So both way, this findings from these three scientists gave us an output that we could treat T regulatory cell specific biology in two different ways. Either we can treat autoimmune diseases, or we can also treat cancer to the patient by just fluctuating the T regulatory cell population. So this is a unique way to harness the biology of T cell, particularly regulatory T cell, in controlling disease. All right, we'll move on and see that what are the interesting thing about the mucosal thing apart from the Nobel Prize of T regulatory cell, and also the things which we have in the human being right now. So there are a huge number of diseases which are happening because of the involvement of mucosal surface. For example, the respiratory problem, the stomach problem, or any kind of a problem in the gastrointestinal tract and the urinary tract. And these are all caused by viruses, bacteria, parasite. Now we don't have any vaccine, so there are requirement of vaccine for those conditions which are there in the human being, or human beings are suffering from that. So some of the very common disease conditions are listed here. So respiratory syncytia virus, which many people are suffering across the globe, and there is no approved vaccine for that. We do have vaccine for some of the diseases, but their coverage is not perfect. For example, Mycobacteria tuberculosis that can cause TB. SARS coronavirus, of course we know, we have gone through the pandemic of SARS coronavirus too, and we know that we do have vaccine, but the vaccine coverage is not sufficient to protect the people from the disease, Streptococcus pneumoniae that cause pneumonia, Bordetella pertussis that cause pertussis, and also influenza-like symptom from bacteria called Haemophilus influenzae. So then these all pathogens are kind of causing a disease condition because the vaccine coverage is now optimum. Apart from that, we don't have any approved vaccine for cancer such as lung cancer, colorectal cancer, stomach cancer, other kind of cancers such as many viral diseases can also lead to cancer. So these viruses are called oncogenic viruses such as Epstein-Barr virus, human herpes virus 8, human T lymphotropic virus 1. So these are the viruses which can cause cancer in the human being. There are viruses which are transmitted sexually such as HIV and hepatitis C virus. Now we don't have any approved vaccine for those to treat patient. Apart from that, we have some of the very important bacterial disease condition in the gut such as Shigella. This is again a very important pathogen of the gut. Enterotoxemia or causing E. coli, that's also a very important bacterial disease condition that causes toxemia in the human being. And then Helicobacter pylori, which can lead to gastric cancer in certain cases when people are susceptible for it. So these are some of the very important disease condition in human that happens because of the problem in the mucosal surface. Therefore, understanding the mucosal immunity is very important. But we do have some very important vaccines which are available in the market to treat those disease condition as well. So let's try to look those finally what are the things which are available in the market that can help us to understand that what are the burden of the disease as well as what are the cure and preventions which we have available in, for which we have some solution. So there are disease conditions such as cholera, which is again caused by a bacteria called Vibrio cholerae, and Vibrio cholerae is actually a pathogen that infect the alimentary canal and cause cholera-like symptoms. So there are vaccines which are available in the market such as oral vaccine, Dukoral, the trade name. These are the name given to the vaccine by the company, but these are actually the heat and formaldehyde inactivated O1 serogroup. So these are specific serogroups of the bacteria. So these are the combination of Inaba and Ogawa. These are the two strain of the Vibrio cholerae which is there in the vaccine formulation. Apart from that, the cholera toxin B subunit. So the cholera toxin is having multiple subunit. The toxin B subunit is actually non-pathogenic in nature, and therefore people are using the cholera toxin B subunit as an adjuvant. That means it can mount and boost the immune response to a better extent. So this is actually a unique combination where we have two strain along with this adjuvant, the B subunit of the cholera toxin. Apart from that, we have the other composition available where we have three strains together, Inaba and Ogawa. These are two important strains, so every vaccine is having that, along with 0139, that's strain from India, that's also included into this cholera vaccine formulation. We do have other where you have only Inaba strain along with the cholera toxin A subunit, which is actually also important form of the toxin, but that's got attenuated, so this won't cause any disease condition. That means these two are inactivated. That means we can use all the heat or the formaldehyde to make it inactive, so bacteria won't cause anything, while in this case this is live attenuated. That means the bacteria can still multiply but won't cause a disease condition because we created a lot of mutation in the bacterial genome, so they won't produce any disease condition. So we are covering the mucosal vaccine because we are talking about some of the interesting facts about the mucosal vaccine, but essentially we will cover all the vaccine aspect in our later part in the last module. What are the different types of vaccine, how they act. Apart from that, we have Salmonella typhimurium typhoid vaccine, which is actually again the combination of different components of the bacteria. So we will not go in detail about all these things, but this is just a outlook of what are the options available in the market which can protect us from different mucosal disease conditions. So these are few of them. There are other viral diseases, so these are bacterial, cholera and typhoid, and the other disease conditions such as influenza, polio and rotavirus. These are very common pathogen in Indian condition as well. So these are also treatable with the vaccine available in the market. So we do have a live attenuated oblique reassortant vaccine. Reassortant stands for the mixture of the two different strains. So we can generate a reassortant virus where the genome can be mixed together. So there are some part of genome from one virus, and the other virus can provide some other genome material. Therefore, this is a kind of a chimeric virus which contain the different cocktail of the genomic information. So this is what you call resortants. So this is created by a process of reverse genetics. So this is a process how we can generate a live attenuated vaccine by reassortment. So this is actually a nasal spray. So one can easily get a spray in the nostril, and the virus can replicate in the upper nostril area and can provide a good immunity in order to have any infection of the influenza coming in future. Although the immunity is not long lasting, so this is actually a seasonal kind of vaccine. We do have oral polio vaccines available in the market that can have different serotypes, and of course those serotypes are all attenuated. So these are actually live attenuated vaccines which are not causing disease condition, although they can multiply, but they won't cause any disease condition. Rotavirus is again a very important disease that caused by fecal-oral route. So if the patient or if the susceptible person gets contaminated food and water which are carrying this pathogen, then the disease can be transmitted to those healthy individuals. And essentially some of the things available for the treating rotavirus infection, particularly in the young ones, rotavirus diarrhea is very common. So we do have vaccine available in the market, such as live reassortant vaccine and also live attenuated vaccine, such as Rotatec and Rotarix. So these are all different kind of nomenclature which are given. So these are actually reassortant virus from the bovine and the human origin, and essentially they can protect a good amount of they can provide a very good immunity to the patient who are actually suffering from rotavirus, and also they can protect from the rotavirus infection. So these are all list of the mucosal vaccine which we have in the market currently. And why we covered this, because essentially we know that the mucosal surface is important. There are disease conditions which are there, and still we are looking into to make more and more vaccine, more and more effective vaccine for diseases which can treat the mucosal surfaces in a better way. So this is all about some of the facts of the mucosal immunology and biology of the mucosal immunology, which we have covered in the module in a great detail. So we'll try to see something else in the next module, and how this immune modulation can lead to other things in some other organ system. Thank you.